everyone, my name is Akemi Shibuya. I'm postdoctoral fellow at INPA, the National Institute for Amazonian Research, and I am member of the Amazonian Freshwater Fish Research Group in this institution. I received my doctoral degree in biology at INPA in 2009, but since my graduation, I have been researching elasmobranch, especially neotropical freshwater stingrays. My interest is to research the feeding habits and functional morphology in elasmobranchs, but also in freshwater teleosts. Beforehand, I would like to apologize for possible mistakes in my English because it is not my mother language. Today, I will talk about a particular sensory system, the lateral line canals. That was one of the subjects of my doctoral thesis, and I published 2 articles about lateral line canals of freshwater stingrays. The title of my presentation is Finding Food Without Seeing: How Freshwater Stingrays Use the Lateral Line Canals in Prey Localization. Probably you are asking who are freshwater stingrays that she will talk about. The neotropical freshwater stingrays belong to the subfamily Potamotrigonidae, and all species live in freshwater environments, not occurring in marine waters. This map indicates where the stingray species were already sampled in South America, and most of them have been reported to Brazil. This subfamily is composed of 38 species in 4 genera, distributed in 4 genera. And presents a diversity of dorsal color patterns and a wide range of body size, with species reaching more than 2 meters width. So, what is the lateral line system? Probably this is the question on your mind after seeing the title of my presentation. We are usually familiar with the sense of sight, smells, taste, hearing, and touch, but some aquatic organisms present different sense organs. The lateral line system is an important detector of water movements near the skin surface. These movements can be generated by individuals of the same species, predators, prey, and the water flux of the aquatic environments. The sensorial unit of the lateral line is the neuromass, a receptor that I will talk more during my presentation. This system is present in some amphibians. Here there is an example of a tadpole of an Amazonian species. The arrows are showing small pores of lateral line arranged along the head and also the body. In fishes, the lateral line is arranged in different forms on the head and trunk. Both species are from Amazonian rivers. The first example is a pacu. Pacu is related to piranha, but unlike piranha, it is herbivorous. You can see that this particular species of pacu presents a well-marked dark lateral line on the trunk. Another example is the electric eels. Actually, they are not eels, but present similar body shape and are capable of generating a very powerful electric discharge. The lateral line canals in electric eels are visible because of deep pores on the skin surface, and arrows are showing pores and marking the position of each canals. But The lateral line system in stingrays presents different forms. Primarily can present three types: superficial neuromass or pit organs, vesicles of Savy, and canal neuromass, which have porous and non-porous canals. Superficial neuromass are located on the skin surface and are exposed to the water. They receive information of the external environment, and especially the water movements very close to the skin. This microscopic image is from a marine elasmobranch, but I'm using that just for illustration. The arrow shows the location of the groove of one superficial neuromass on the skin, protected by two placoid scales. Another type is the vesicles of Savy. These vesicles were first observed in electric rays. They are found under the skin and there is no contact to the external environment. Each vesicle is a pouch with neuromass, and in the case of electric rays, we can find two rows on the dorsal region, and lots of them near to the mouth on the ventral region. But we can also find some vesicles of Savy in stingrays. Here we have an example of marine species, and there are few vesicles arranged in rows near to the mouth. Canal neuromass is situated under the skin and can be found in both dorsal and ventral surfaces of the body. These canals are filled by neuromass arranged in a single row. The porous canals have tubules that terminate in pores opened on the skin surface. That means neuromass of this type of canal have direct contact to the external environment. And ventral canals can present sections porous, here with a dashed circle that have similar morphology of those I explained before. And in blue circle, I'm showing non-porous canals, and the neuromass have no contact to external environments. In short, superficial neuromass and porous canals have contacts to the external environment, and vesicles of Savy and non-porous canals do not have any contacts. After reading many studies of sensory system in marine species, I asked myself, what are lateral line canals of freshwater stingrays like? This was one subject of my doctoral thesis. And the main goal in examining the lateral line canals was try to answer the question, does the lateral line system guide the stingray to locate and capture its prey? And now, first I will explain the field collection and how I obtained these specimens. The study area was the Middle Rio Negro Basin, a blackwater river in the Amazonas state, Brazil. Stingrays were collected in streams, lakes, and small rivers of Barcelos municipality. These pictures are examples of where usually stingrays live: in shallow waters, igapós. Igapó is the flooded forest in Blackwater River, next to sand beaches, and also stingray can be found in the main channel of the river. Basically, we use two main fisher gears to collect stingrays. The bottom long line, as explained in this figure, we use pieces of fish as baits. These two pictures showed my colleagues preparing the hooks and putting the long line in the water. In this video, my friends Danilo and Eudes, one of the best fishermen in Barcelos, were collecting stingrays using rapichet, a deep net specifically used to collect stingrays in rivers. All stingrays were maintained in shelter built with nets on the beach or in plastic box with up to 2 individuals per box. Plastic boxes were also used for transportation from the field to the lab. These are the 5 examined species collected in Rio Negro: the ocellate stingrays, Potamotrigon motorum, smoothback stingray, Potamotrigon urbignae, rosette stingray, Potamotrigon schroederi, cururu ray, Potamotrigon wallacei, and discus ray, probably another species of paratrigon. All individuals were euthanized to be later fixed in formalin solution and preserved in 70% ethanol solution. It's important to say that the euthanasia of stingrays was conducted in accordance with the Ethical Committee of Animal Experimentation. So, to answer the question, does the lateral line system guide the stingrays to locate and capture its prey? We also used previous information about diets of those 5 species collected in Rio Negro Basin. Information of the diets were also published by myself and my colleagues, and shows that freshwater stingrays feed on fishes, crustaceans, basically crabs and small shrimps, and insects, most of them dragonfly larvae. Each stingray species presents different dominant prey, except Potamotrigon wallacei, which presents a generalist feeding habits. And all prey categories lived buried in the litter and sand substrate, and all identified fishes lived associated to the bottom. These were the most common prey items in the stomach's contents. They were partially digested, but had relevant undigested characteristics to identify up to the level of family of each prey category. As I said before, stingrays feed on dragonfly larvae, shrimps, crabs, and fishes, most of them small fishes. In 2010, I described the distribution of lateral line canals in 4 species from Rio Negro. In this publication, I also quantified the neuromass in Potamotrigon motorum and Potamotrigon urbignae. That was the first publication relating the morphology of lateral line canals to the feeding habits in freshwater stingrays. For examination of the organization of lateral line canals, skin was removed from both dorsal and ventral surface, and all canals were reviewed. Many studies have described the functional difference of porous and non-porous canals. Porous canals on the dorsal surface detect movements near the skin, such as water flow of the river, as well as can identify the presence of potential prey swimming on the bottom. Pressure of external environment felt by neuromass, which send this information to the process center in the hindbrain. But in case of porous canals of the ventral surface, their function is to identify movements of buried prey in the bottom, or those prey living on the substrate. A non-porous canal have an important tactile role stimulated by touching the prey on the bottom. My questions to study the lateral line canals was, how does stingray feel the buried prey when touching? Firstly, to quantify the neuromass, all canals were mapped after remove the skin. A blue dye was injected in multiple parts of all canals, and then they were opened to expose the neuromass, and they were counted under the microscope. This drawing shows the arrangement of the neuromass in a single row inside the canals. And in this picture, arrows are indicating each neuromass. Actually, what you are seeing is the gelatinous cupola of the neuromass. Is it hard to see? And now, can you see? I delimited each neuromass in yellow and the canal walls in red for easier visualization. Now you have an idea of what is the neuromass. To understand the organization of neuromass in non-porous canals, pay attention to the left representation. It shows the position of the cupola of neuromass inside the canals, and neuromass are indicated in this drawing and in this picture. Above the cupola of neuromass, you can see the canal wall, and above that, the skin surface. The right figure shows how stingray can feel the prey by tactile stimulus. Blue arrows in opposite directions indicate the flow of canal fluid responsible to stimulate the neuromass after touching the prey. An interesting characteristic of porous canal is the presence of tubules that terminate in pores on the skin surface. Those drawings are showing two different tubules of the dorsal and ventral canals. An interesting characteristic of porous canal is the presence of tubules that terminate in pores on the skin surface. Those drawings are showing two different tubules of the dorsal and ventral canals. The lack of neuromass is visualized in this picture. You can see the presence of neuromass in the canal, indicating by black arrows, and the orange arrows showing the tubules with no neuromass. In general terms, tubules are connection between canal and external environment via pores on the skin surface. I call here PM for Potamotrigon motoro and PO to Potamotrigon urbiginae. And N represents the total number of neuromass, and D is the density of neuromass. The density of neuromass is the mean number of neuromass per centimeter of canal. I divided the body in three parts: anterior, central, and posterior. PM for Potamotrigon motoro and PO for Potamotrigon urbiginae. And N represents the total number of neuromass, and D is the density of neuromass, that is the mean number of neuromass per centimeter of canal. And I divided the body in 3 parts: anterior, central, and posterior. For the dorsal surface, both species have similar number and density of neuromass in each part of the body. This balanced distribution reinforces that dorsal canals are responsible to identify water movements on the entire body surface. But the highlights of my results are on the ventral surface, with the very large number of neuromass in both species, as well as the density of neuromass for anterior parts of the body. These results shows the position of neuromass on the anterior parts of the ventral surface reinforce the tactile importance for accuracy of prey detection and strike during benthic feeding behavior. After finish the first publication, I have this question: how is the distribution of neuromass in the ventral lateral line canals in other freshwater stingrays? Especially because the huge difference of canal organization on the ventral surface for paratrigon comparing to potamotrigon species that I had observed during my doctorate. 10 years after the first publication, I published this article. You probably think she spent 10 years to examine it and publish the other 3 species from Rio Negro. My answer is yes, I did. But the reason is it's really difficult to obtain specimens with reasonable size for neuromass quantification under the microscope. And I avoided neonates and very small juveniles or very large adults. In this publication, I emphasize the distribution of ventral canals for potamotrigon schroederi, potamotrigon wallacei, and paratrigon. Comparing the ventral canals between these 2 groups, potamotrigon and paratrigon, basically potamotrigon presents canals surrounding the mouth, and paratrigon has a web-like distribution forming a tangle around the mouth. I followed the same methodology of the first publication to quantify neuromass and calculate the density of neuromass. Here I present the number of neuromass and densities for each species separated in anterior, central, and posterior parts of the body. For all 3 species, the highlight was the anterior parts of the body with a high number of neuromass. But paratrigon has an incredible high number of neuromass comparing to potamotrigon species. However, considering the density of neuromass, paratrigon had similar densities for anterior, central, and posterior parts of the body. On the other hand, the high number of neuromass in the anterior part increases the sensitivity of mechanotactile receptive field around the mouth. The specialized web-like organization in paratrigon possibly compensates the similar densities of neuromass among parts of the body. The high number of neuromass in the anterior part of the body for all species clearly confirm the use of non-porous canals to identify tactile stimulus generated by buried and substrate-associated prey. In short, I have to say that obviously pre-localization in freshwater stingray is not carried out using solely the lateral line canals. Other sensory systems, olfactory, electroreceptive, visual, and auditive systems are acting jointly during foraging. However, the complex organization of the ventrolateral line system in freshwater stingrays with porous and non-porous canals provides an increased capability to detect prey in a wide range of substrates types. This was my presentation. I would like to thank the Erasmus Week organization for this amazing invitation, and I hope you had enjoyed my presentation. Thank you.